This segment of your notes is on meiosis, or gamete cell division. Pay close attention to that word, meiosis. The correct pronunciation, it looks like it might be meiosis, but it is actually meiosis. And it is a sister process to mitosis. It just makes a different type of cell. So first of all, let's talk about the amount of DNA in gamete cells, and we'll get to what somatic and gamete are here. So a somatic cell is a cell that is found anywhere in the body. So if you look at your diagram here, you have an egg cell that is fertilized by a sperm cell, and that begins into somatic cell division. That would be body cells, cells of your skin, cells of your muscles, cells found anywhere in your body except for your gametes or sex cells. So a gamete is a sex cell. A somatic cell has a full set of DNA. A gamete cell has, or a germ cell has, half of the DNA. And there are terms for those amounts of DNA, and they are diploid and haploid. Diploid means that it contains 2 copies of each chromosome, so one from mom and one from dad to make 2 copies. So there's a copy of your mom's DNA and a copy of your dad's DNA in every cell of your body except for your gametes or your sex cells. Your sex cells are haploid, meaning that they have 1 copy of each chromosome. They are haploid in order to become diploid after fertilization. So you take half and half and make one whole set of DNA, and then that one set of DNA is duplicated. Little overview of meiosis, this whole process. It looks very, very, very similar to mitosis. And the only difference being is that it goes through the cycle twice. There are some other little subtle differences that we're going to get to, but the biggest primary difference is that it goes through cell division twice. And you'll see that it has those phases PMAT, just like mitosis did. It just has them twice. Meiosis I divides into 2 diploid cells. So we take the cell, we duplicate the DNA, we make cells like we do in mitosis. And then meiosis II divides the cell into 2 unique haploid cells. The end result of meiosis II is 4 cells, because if you take 1 cell, divide it into 2, divide those 2 cells into 2 more cells, you have a end result of 4 daughter cells from 1 germ cell. Alright, so the first phase of meiosis is interphase. Interphase is very similar here as it was in mitosis. The cell is going to increase in size, replicate DNA, and prepare for division, and make RNA and proteins during this kind of growth in normal state time. Prophase I, the nuclear membrane breaks down, so that should sound familiar to prophase and mitosis. The chromosome pairs are going to group together, so this looks a little different than what you see in mitosis, but you have the pairs of chromosomes pairing up and grouping together. And then this is what makes meiosis unique. During prophase I, crossing over happens. So I've got another picture on the next page that I want to show you of what crossing over is. So the chromosome pairs have, the homologous chromosomes have lined up next to each other. We've got 1 chromosome with the centromeres here. And when they line up together, they cross over, literally cross the segments of their DNA over and exchange those DNA segments. So you see here we started off with blue on the left, red on the right. And then in our final picture, we have some that have recombined. In the middle, they're called the recombinant chromatids. And on the outside, they're the non-recombinant chromatids. And so what that's saying is they have different segments of that portion of DNA on those recombined chromosomes. Now, this is really important in meiosis to give us that genetic variability. So if you have siblings, you know that you all generally look different. For example, I have triplet siblings, and so they were all born at the same time. You would think they look alike, but they don't. One of my brothers has black hair and brown eyes, one has blonde hair and green eyes, and my sister has brown hair and brown eyes. So they don't look anything alike, and that's because of this process of meiosis that creates genetic variability. And in the next unit in January, we will get into why that is and some more of how that happens. How does one have blonde hair and one have brown hair? That can't be possible. But we'll get into all of that when we go through the unit on genetics. But please remember, it comes from this process of making sex cells and crossing over in prophase I. All right, on to metaphase I. So in metaphase I, those same chromosome pairs line up in the middle of the cell. Remember, meta means middle in this context. In anaphase I, the chromosome pairs are separated to opposite sides. So this again is familiar. Ana means pulled apart. AA. Telophase I, we also have cytokinesis that happens here with telophase I. But in telophase I, we have 2 new cells created. And so we have 2 cells for the first time at this point. So on your note sheet, you should see a blank in each of your boxes that says number of cells. And up until telophase I, you still only had 1 cell. Now going into meiosis II, we're going to have 2 cells at every point in that stage, except for telophase II. We're going to have 4 cells. So telophase I, we have 2 new cells totally created. We've divided our cytoplasm just like in cytokinesis in other instances. On to prophase II. During prophase II, the nuclear membrane breaks down, and we begin that whole process of division again. We have spindle fibers forming and the centrioles moving to the poles, just like in prophase of mitosis and prophase I of meiosis. During this phase, we do not have any crossing over occurring. That already occurred in prophase I and does not happen again anywhere else in the cell cycle. Metaphase II, this is another metaphase, so you should be able to already guess that they're going to line up in the middle of the cell. So the chromosomes line up in the middle of the cell. And pay close attention to your diagram here. You'll see that you have your chromosomes, but they have, so like this red chromosome has a little blue tip on the end. Well, that's because of that prophase I and that crossing over that happened in prophase I. So at the end of this process, we'll look at the colors and see how maybe they've changed from what you anticipated. Anaphase II, we have the chromosomes or the sister chromatids are pulled apart. So that single spaghetti noodle wrapped around a fork, they're pulled apart by their centromeres. And in telophase II, our nuclear membrane reforms, and we have 4 haploid cells created. So these all have half of the genetic information. So pay close attention here. This cell at the top here with the red and green, it doesn't have any recombinant DNA in it. But the one at the bottom has some red with a little tip of blue and some green with a little bit of purple. that is a recombinant haploid cell. So all 4 of these here in your picture are unique cells. They are all different from each other, and as an end result, the offspring may be different. Here's a quick diagram for you. So mitosis is attack of the clones. You have one cell that divides into 2 that are identical to the beginning. Meiosis is the opposite. They are different cells. So you start with one cell, it makes 2, those 2 make 4 gametes that are different from each other. So we have 46 chromosomes in the beginning in meiosis and 23 chromosomes in the end. So you'll see here when it starts to count 46, then we have 23, then we have 23 in the offspring. Okay. And then mitosis, we start with 46, we end with 46. And that concludes your mitosis and meiosis notes.